talk of algorithm session and also of JOBIM for this session is Cédric Chauve, who is, well, is it super early for you, Cédric, today?
It's 2 in the morning.
Yeah, wow. Okay, that's
fine.
brave.
Okay, so I can start to share my screen. Okay, so thank you very much for letting me present our work on isoform discovery using long-read sequencing. It's a work that has been present or will be presented at ReComb, and there is a preprint available on bioRxiv, and we're working on the journal version. So about isoform, I want to make the distinction to start between two problems. The first one is identification, where you have a database of known isoforms in that slide ABC. And from long reads transcriptomics data, you can detect, for example, on this example, that A and B are present, and there seems to be a third isoform that is not in the database. So this is identification. And we are interested in discovery. That means that we want to be able to discover that there is that fourth isoform that was not in the database or it was not known a priori and is present in the data set. So Freddie, the tool I'm going to present, is a de novo isoform discovery tool. That means that we don't rely on any kind of database or annotation to start with. So obviously, long-read sequencing is a very attractive technology, especially for isoform identification, due to the potential to have full-length transcripts in a single sequence molecule, even if some isoforms might be too long for that. But it's actually not as trivial as it might look, especially in the case where we work with no annotation and no database, because the high sequencing error rate makes it difficult to detect exons. And there can be actually also sequencing structural artifacts in the long reads data from transcriptomics. So I'm going to describe the method we developed to address these issues. So the tool is called Freddie, and to be honest, I don't remember why that name was picked, but that's what it is now. And it works in 3 steps. The first one, actually 4 steps. One of them is kind of hidden here. This is the alignment of the long reads, the transcriptomics long reads onto a reference genome. So we assume that we have an assembled genome that we can align the transcriptomics long reads onto. And we use for that Minimap, a splice alignment mode. Then you split the genome into intervals based on the long read coverage. That's quite usual. Given that we work with no isoform annotation, the second step is actually very important. We split these intervals into what we call canonical exons that are supposed or assumed to be exons of genes. So if you work with a database of known isoforms, you have exons coordinates, and that makes it much easier. Here we don't have exons coordinates, so that step identifies potential exons. And from there, we are going to encode the long reads into a binary format based on these canonical exons, and we're going to cluster the long reads into groups that each correspond to one potential isoform. So the segmentation, the first part, intervals, is very simple. We just partition the genome into sets of intervals such that no 2 sets are linked by any read alignment. So a picture is better than words. You can see here that we have 3 kind of intervals, 1 and 2, based on the coverage by long reads. So that means after that we're going to be able, for the second phase where we identify the exons, to process each of these segments, these intervals, independently. So that is very simple. Then now if you take here an example with some underlying isoforms and the intervals, then this is an idealized example where the canonical exons are very easy to identify. But that's obviously an idealized example, and this is not what we observe on real data. Okay, so what we do for the segmentation, actually, for each position of an interval, we count the number of reads that start or end an alignment in that interval. And that gives us these peaks that tells us, how many reads start an interval at this position and so on. And the data you obtain like that is a little bit jagged. So we smooth it with a standard Gaussian filter. And then we try to identify now what would be the boundaries of canonical exons. And we look at the peaks that are places where we see a difference in coverage by long reads. So here we have 6 peaks, and the peaks 4 and 5, they look a little bit dubious. And indeed, that's an example that was actually taken from data. They don't correspond to boundaries of canonical exons, but how do we define that? Then we use a dynamic programming algorithm that is going to identify that these two peaks are not canonical exon boundaries. And for that, we define a scoring function that I'm not going to get into details, but basically penalizes reads that only cover partially one segment, and gives a positive score for reads peaks that show a sharp decrease in coverage on one side of the peak. That identifies that a bunch of reads suddenly stop to cover that segment and then start to cover the following segment, or conversely. That's a simple scoring function that is available to dynamic programming with a cubic time algorithm. And we've been working lately on a bunch of optimization to make it faster. But once we've done that, we identify in that example 4 boundaries for canonical exons. And once we have these canonical exons, then we can just encode the long reads into a binary matrix with a 1 if a read covers a canonical exon and a 0 if it doesn't. So now we have very nice data to work on an optimization problem. And the next problem will be to cluster these reads into groups that are assumed to each originate from a single isoform. So the problem is that we don't know how many clusters we start with. This is unsupervised because we don't know how many isoforms there might be. And we don't want to overfit that, basically put every read into its own cluster. So to do that, we designed an optimization problem, the minimum error clustering into isoform problem called MERCI, or MERCI in French. And how does it work? We have a start of reads, a set of reads to start with, and we're going to split them into 2 groups. One is an isoform bin that will represent one isoform, and one is a recycling bin that we're going to use in the next iteration of the same problem. And how do we define how we put reads into the isoform bin? Then we're going to minimize the number of 0 in the binary matrix that we want to turn into 1 in order that all the reads in the isoform bins are consistent with the same isoform. So it's actually completely inspired by haplotyping with long reads, where you want to correct also potential errors in the long read data. So it's a relatively, so the principle is simple, obviously, with the power of, we solve that with integer linear programming. So that gives us a way to define much more refined constraints for the problem. But the principle is simple. We want to minimize the number of zeros that we turn into ones in order that all the reads in the isoform bin agree with the same isoform. Okay, so, and in the same time, we want to maximize the number of reads that we can put in the, minimize the number of reads we put in the recycling bin. So that will work like that. We will first identify one isoform, and then the recycling bin, we're going to process it again and obtain 2 other isoforms. So, and And we solve that, as I said, with integer linear programming. So just really very standard optimization techniques. So let me present some results on simulated data. And we compare with two tools, FLAIR, that uses an annotation database, so database of non-isoforms and exons, and string-tie2, which is fully de novo, so comparable to Freddie. So the first thing we look at, this is on our simulated data, do we identify properly exon boundaries? So obviously FLAIR with the full annotation database does best, but you can see that when we compare string-tie2 and Freddie, we do significantly better than string-tie2 to identify precise exon boundaries based on the true exons, okay, because these are simulated data. It might look a little bit like a small improvement, but actually this is a different scale on the top. And so we do identify a much larger number of exact exon boundaries. Then if we look only at the predicted exons, we can see that the three tools do quite differently with string-tie2 here that has a lot of exon that it is the only tool to predict, contrary to FLAIR, which is that column here, Freddie, which is that column here, and there are almost 7,000 exons from the ground truth, or sorry, 5,000 exons from the ground truth that are not detected by any tool actually. But what is interesting, this is that column that shows that Freddie identifies quite a lot of exons that are true exons that the other tools do not identify. So when you look at that picture, basically it shows that Freddie and FLAIR do relatively well on this data, actually quite well, and string-tie2 does a lot of things that it's the only tool to predicts a lot of exons that no other tool, including the ground truth, do predict. And last, so that figure is unreadable, and I'm sorry about it, so I'm just going to give a summary of it. So if we look at the isoforms themselves, so from Freddie, we can get a consensus sequence for each isoform based on the long reads that are in each cluster. And what that figure shows us, this is that Freddie does slightly less well than FLAIR when we use the full annotation database of FLAIR. But Freddie does much better than FLAIR if we use only half of the annotation database chosen randomly, and Freddie does much better than string-tie2. So in a sense, it tells us that even compared to an annotation-based tool, if we remove half of the database of non-exons and isoforms, then Freddie will do much better than this tool, even if it works with no annotation database at all. And we used Freddie on real data that was prostate cancer cell line, the 22RV1 cell line. And we did find two expressed novel isoforms in that prostate cancer cell line. And here is an example of one of these isoforms, and the blob on the top of the chart actually shows a coverage by short reads that we used to as a mean of validating, or at least starting to validate this potential novel isoform. And you can see that the novel isoform here is kind of a mix of two non-isoform, AR204 and AR201. And AR is the androgen receptor gene, which is a very important gene in prostate cancer pathology. And that's it. Let me acknowledge the people I worked with on that. This is mostly the work of Barao Rabi, which is a very talented graduate student at UBC, and that was a work done with my colleague, Dr. Farazak from UBC. Thank you, and I'm ready to take your questions.
Thanks, Cédric. Let's see if there are questions from the audience. I will maybe briefly ask a quick filler, which is, I'm confused about the fact that you showed that string type had a lot of exons, which seems to be exclusive to it. And in the next slide, you evaluate false negatives, but we don't know really if those extra exons are false positives.
Also, the next slide, it was not exons, it was full isoforms. So based on the sequence of the isoform, not just the exon coordinates.
Okay, so this clarifies half of my interrogation. The other half would be like, is the exclusively predicted exons from string type likely true or likely false?
Likely false, because it's simulated data, so we know which were the true exons. So it's not likely false, it's completely false.
Excellent, thanks. A question from Nawal Mansouri, who asked, hello, which data was used to perform AR isoforms?
It was the 22RV cell line. It was, yes, that's a good point. Oxford nanopore sequencing of the 22RV1 cell lines, and it was not direct RNA, but cDNA, transcriptomic sequencing.
Okay, nice. A question from the third room of Montpellier, who asked, do you find RNA fusion boundaries?
No, not with that tool. That's actually another paper of our group that is being finalized right now to detect gene fusion from transcriptomic long reads data.
Sounds good. A question from Laurent Jourdain, who asked, do you plan to join the LR GASP challenge? It is a dedicated challenge on de novo annotation.
I didn't know about that, but then I will definitely look into that. Yes, that sounds interesting from the name.
Does, yes. A related question on me is, recently there was a complete sequence of a human genome, which was pre-printed by the telomere to telomere consortium. And in that paper, I checked they use string type 2. Can you comment on whether they did the right thing or they should use maybe Freddie?
I think it's too early to claim that they should use Freddie. These are simulated data. And we are working actually very hard to compare the tools on real data, but for isoforms annotation, for isoform discovery, it's much more difficult to define which tool is better than which other tool because of the lack of ground truth.
Yeah, that makes sense. So actually, to understand the focus of Freddie more precisely, in your title, it says detecting alternative splicing. It seems that it can also create annotation from scratch.
I don't know. I mean, we don't use annotation, but what do you mean by create annotation?
Like if you want to create a GFF file for a new assembly.
Oh, I didn't think about that.
Oh, that's okay. A question from Thomas Derien, who asked, did you try to evaluate Freddie performance with repeat associated transcripts?
No, no. That would be a good idea, actually, to focus on that. That might be a good summer project for this summer. That's a very good idea, actually.
Inspirational question. Another one from Laurent Jourdain, who asked, string type can also be used with annotation, I guess existing annotation. Did you compare your tool with this version of string type?
No. The focus was really on fully de novo.
Makes sense.
But we will eventually, because we're going to work on a version of the tool that actually also uses annotation. Okay. So eventually we'll do that.
Yeah, nice. Brief one from me. So is this applicable for both IsoSeq and nanopore?
Yes.
Okay.
Yes.
Excellent.
You just need long reads.
Excellent. Okay, so I've been bombarding you with questions. Nice job with the quick answers. Thank you. Let's see if there is a last minute one from the audience. And well, if not, thanks again, Cédric, for this talk. especially giving
Thank you.
At 2 a.m. your local time.
Yeah, I'm back to bed now.
Let's thank all the three speakers from this session again. So Anne, Pierre, and Cédric, and this concludes the algorithm and data structure session 2 of JOBIM. Thanks for your attendance.